let's go ahead and continue on with our list of amino acids. The one I want to do now is sort of an honorary member of multiple categories. So this one sort of fits in the aromatic category. It also fits in the charged category, and it also fits in the polar but neutral category. So I don't know necessarily where to put it, because you'll see that it has character that is consistent with sort of all of those areas. And that is, and it's just one, that's the amino acid histidine. Now that word probably sounds familiar to you because you've heard of histamine. You probably, if you have allergies, you've probably gone to the store and bought antihistamines. Histidine is the precursor to the molecule histamine, and we'll talk about its synthesis probably next time. And its one-letter code is H, so that one at least makes sense. So the backbone structure is the same for histidine as it is for the other amino acids. On the you've got a side chain on the alpha carbon. It's a dash. Then here's the beta carbon, and the beta carbon is bonded to a ring. But the ring, and I think you've seen this before because we talked about the catalytic triad, and you've dealt with a ring like this before on the what is the charge on that molecule. The ring has this basic structure, and that functional group is called a 5-membered ring with 2 nitrogens arranged in that form is called imidazole. It's aromatic, and it's also polar because it's capable of hydrogen bonding. And in the current form, it's neutral. So you can see how it fits into the aromatic and polar but neutral category. However, the imidazole side chain is the conjugate base of a positively charged acidic form, which you've seen before, but which I'll draw for you here. Lone pairs on the nitrogen can pick up a proton to give you this positively charged form. I'll use a squiggly line. Here's the beta carbon, there's the ring. The positively charged form is called imidazolium. If you wonder what a positively charged thing is and you add a ium to the end of it, you'll probably be pretty close. Imidazolium. And the pKa of that positively charged proton, I guess I should have done that one, is 6. So, interestingly enough, at pH 7.4, you expect it to be around 90%, that's not a percent, that's a degrees, 90% neutral, but about 10% positively charged. So that's why it's weird for histidine. You can't put it neatly into, oh, it's always positively charged, or oh, it's always neutral. No, it's actually at physiological pH, it sort of spans categories. You've already seen that matter in the catalytic triad, where histidine goes from neutral to being positively charged as it deprotonates another nearby group. So you can fit in histidine either in the aromatic category, you can fit it in the positively charged category, or the neutral but polar category, so long as you remember that at pH 7 it spans the line between charged and neutral, meaning about 90% of histidines will be neutral and about 10% will be positively charged. Yeah?
So since it's like that, it's close to pH 7.4, would, if, like, for example, if you were to give us a question like what's the charge for this,
No, I would say to answer questions, because it's 90%, generally greater than 50/50, we'll say for what's the charge questions, at pH 7 you're going to consider it neutral. As long as you remember that around 10% is going to be positively charged. Of course, in the stomach at pH 2, it'll be positively charged. Yep. Okay, good. So then let's just do the sort of boring neutral but polar. There are a few in this category. The simplest is probably serine, S-E-R-I-N-E, 3-letter code S-E-R, 1-letter code S. That one at least makes sense. Same backbone as the other amino acids. It's just like alanine, only it's got an OH group on the beta carbon. Of course, the pKa for an OH group that's just normal and not adjacent to a benzene ring is 16, but it is always neutral at physiological pH. We've seen it get deprotonated in the catalytic triad. But in general, if exposed to water, it should be neutral. It's polar, it's hydrophilic, it's capable of hydrogen bonding. All right, so the next one is threonine. Threonine is almost exactly like serine, only it's going to have an additional methyl group on the beta carbon. So if we were to just copy and paste the structure of serine over here, all we'd have to do is add a methyl group to get threonine. The pKa of that proton on the alcohol is still 16. So still neutral at physiological pH, like serine, capable of hydrogen bonding. Threonine is one of these beta branched amino acids. It's but the branching point is an OH group. So I don't know that this counts. Well, I'm going to erase that well, I'm going to leave it there, but I don't actually know off the top of my head whether threonine counts as a beta branched or a branched chain amino acid or not. Sorry, I think it doesn't because the OH group is easily removed, but that's neither here nor there. Both of these are neutral at physiological pH, but they're polar, and so they're comfortable in water. When deprotonated, they could be nucleophiles. So you see that in the catalytic triad, the serine OH gets deprotonated, and it acts as a nucleophile to attack an ester. Go ahead.
Is the serine and threonine also aromatic?
No, they're not aromatic because they lack the ring and the double bonds, and they're not charged because they're neutral. All right. Which ones have I forgotten? All right, there's another one called that you've encountered before a little bit, or I've alluded to it before, CYSTEINE, or cysteine, 3-letter code CYS and 1-letter code S. Cysteine is like serine, only the oxygen has been replaced with the atom that's one row down in the periodic table from oxygen, and that is sulfur. The pKa of this thiol, that's the functional group, is 8. So at physiological pH, you would say it's greater than 50% neutral. And so we're still for what is the charge questions at pH 7.4, the answer is going to be neutral. Of course, if the pH was greater than 8, we would say negatively charged. sometimes cysteine will act as a nucleophile in an enzyme active site. You've seen a couple examples of that in some of the mechanisms that we've talked about this semester. In particular, or this term, whenever we've done something like this, a sulfur attached to the enzyme, that's what we've been referring to. The conjugate base of cysteine is this negatively charged thiolate, and cysteine's a better nucleophile when it's deprotonated. So often the first step of a mechanism is for a base to deprotonate the cysteine side chain, and then it goes and attacks an electrophile. One other interesting thing about cysteine that we won't, that you'll see in selected examples, so you need to be prepared for, is cysteine can be oxidized to form what are called disulfides. In a disulfide, you saw a disulfide before in lipoamide. In a disulfide, one cysteine side chain is, the sulfur from one cysteine side chain is linked to the sulfur from another cysteine side chain via the two sulfurs have a covalent bond. That's a disulfide bond. And cysteines are used all over the place in proteins to make cross links between amino acids to help hold proteins together in a particular shape. The place where you most likely, there's a lot of disulfide bonds in antibodies, in proteins that are exposed to oxidizing environments like hair and skin and fingernails. The place where you most likely have heard of or encountered cysteine disulfides has possibly been if you've ever gone to get a perm. I know that's a thing of the 80s, so maybe that's not a thing anymore. But if you for example have straight hair and you want it to be curly, you can go to the hairdresser and they will curl your hair, and then they'll apply a chemical that reduces your disulfide bonds in your hair from disulfide to cysteine. So the process of going from cysteine to the disulfide is called oxidation. The process of going from the disulfide back to cysteine is called reduction. They'll first reduce the disulfides in your hair, then they'll reoxidize them with the new shape, and then your hair will keep that particular shape for a while. Never done it, don't know that I necessarily recommend it, but yeah. I'm remembering a scene from The Office where Michael Scott was wishing that, I think he was meditating on what the word boss meant and how boss is a compliment. Like, dude, that perm is so boss, which I think hasn't actually been said since the 1980s, so whatever. Okay. I'm also realizing that I think The Office ended 20 years ago, so that's probably way past your time too. No, not 20 years ago, maybe 10. Yeah, somebody can look it up. Yeah. All right. Two more polar but uncharged amino acids. These are related to aspartate and glutamate, so it's going to be really easy to confuse them. Asparagine has the 3-letter code ASN and the 1-letter code N, so there's some way to remember it there. This is the amide version of aspartate. In other words, in asparagine, we convert the carboxylate side chain of aspartate into an amide. So alpha carbon, beta carbon, gamma carbon. In aspartate, it was a negatively charged carboxylate. Here, the negatively charged oxygen has been replaced by the NH2. Okay, and this is again polar but neutral. It's capable of hydrogen bonding, but it's not charged. And we don't, the pKa for the side chain is high enough that we don't worry about it losing a proton and becoming negatively charged. The other is called glutamine. This is the amide version of glutamate. 3-letter code GLN, 1-letter code Q, because we got to use Q for something, and we run out of other letters of the alphabet that make sense. So you're just going to have to memorize that. Again, glutamine is the amide version of glutamate, and it will look exactly the same as glutamate, only the carboxylate will be replaced by an amide. Glutamine is like glutamate, 1 carbon longer than asparagine and aspartate. N and Q. So you have to remember the difference between D and N. D is charged, carboxylate. N is neutral, amide. E is charged, carboxylate. Q is neutral, amide. And so keeping that straight can be a bit of a challenge. That's a place where people get tripped up. I think we only have 2 more at this point. I think I haven't, and if somebody wants to count, I think you've already seen glycine, which I will now, yeah, we'll just put that in our weird category. The weird ones. Glycine, or GLY, or 1-letter code G. And this is weird because there is no side chain. So no stereocenter, no side chain. It's not particularly hydrophilic or hydrophobic. It's maybe comfortable in all of those environments, but importantly, it is way more flexible than the other amino acids. We're not going to talk a lot about this. I would love to. This is a big thing to me, but we're not going to say anything about it. Amino acids, at least in the backbone, can rotate around each of these 2 bonds. If you have a side chain, if you could imagine back at the beginning of the term, we talked about Newman projections and keeping the biggest stuff anti versus gauche. If you have a side chain at one of these 2 positions, the dash position, for example, it changes which shapes the side chains prefer to adopt. If you don't have a side chain, those bonds are much more flexible in terms of rotation. So there are many more allowed conformations of glycine than for the other amino acids. The other weird one is called proline. Proline is weird almost for the opposite reason as glycine. Proline is way less flexible. And the reason proline is way less flexible is that the side chain of proline actually ties back and is bonded to the backbone amino group. So this is proline, and that's the alpha carbon, the beta carbon, the gamma carbon. Proline's got a 5-membered ring, which actually makes rotation about the N to alpha and alpha to C bonds in the backbone restricted, or more restricted than would otherwise be. Proline is found in a lot of places, but especially found in collagen. And proline in collagen actually gets modified with an OH group on one of these to form hydroxyproline. You may have heard of proline in collagen peptides before. Okay, so is that 20? Did we do all 20?
Yes.
Did we really, or you just want to stop? I think that's it. I'd feel bad. It's like I'd forgotten one of my children if I forgot one of the amino acids. Okay, there you go. So there's the 20 amino acids. You need to know their structures, their names, their 3-letter and 1-letter codes, and then just something about their general character. The categories mostly should do it. Polar, but neutral, charged, nonpolar, and so on. All right, now we'll talk about how the amino acids are metabolized. But first, I want to talk about what a peptide or a protein is. We're not going to talk about yet about how peptides and proteins are biosynthesized. That's going to come later next week. But for now, peptides and proteins are polymers of amino acids where the amino group of one amino acid is linked to the carboxylate of another via an amide bond. So let me draw for you a tripeptide. The sequence of this tripeptide is A, A, A. That means I should have an alanine amino acid with a methyl group side chain. And then this is the carbonyl carbon of the first alanine. And it's going to be linked to the next one via an amide bond, which I'm going to draw in blue. Okay? And then that nitrogen belongs to the next alanine. Now, because I've drawn this next alanine with the nitrogen on the left, the carboxylate group on the right, and the alpha carbon pointing upward, the side chain should be a wedge rather than a dash, and it's still a methyl group. And then the connection between this alanine and the next one, I'm drawing in blue. That's also an amide bond. And then we can draw the third alanine. And then the third alanine, that's the end of our peptide, so this is just the carboxylate and should be negatively charged. These blue bonds are bonds between different amino acid subunits. So for example, let's just highlight this one as pink. So this is the first alanine residue or amino acid in the protein. The next one is orange. This is the second alanine. And then the last one will be like blue. This is the third alanine. So when you look at a peptide, you can divide it up into its component amino acids, which they're no longer amino acids anymore. They're involved in amide bonds. So we'll call each one a residue. For example, the orange one is the second residue, the second alanine residue. The blue bonds are functional groups the blue bond is between the carboxylate carbon of the first alanine residue, the pink alanine residue, and then the nitrogen of the orange alanine residue. And what I've highlighted with the dotted gray line is what we would call an amide bond, which is used interchangeably with the phrase peptide bond. Okay, so there's an amide bond there. There's an amide bond over there. For a tripeptide, you only need 2 amide bonds. Notice that the ends of the tripeptide are different from each other. On the left, the pink alanine residue has an ammonium group, whereas the blue alanine residue has a carboxylate group. We call the end of the peptide that has an ammonium group the N terminus, where N is for nitrogen. We call the end of the peptide that has the carboxylate group the C terminus, where C is for carboxylate. We can draw the peptide in any orientation we want, but when we write the sequence, we always write the sequence. It's always understood to be written from N to C, always. Sequences are always written N to C. Now with this alanine tripeptide, there's no possible confusion because they're all alanine. But what I want to show you real briefly is that the peptide AEY is different from the peptide YEA. Oh, I didn't even do that on purpose. Yay. Okay? So I want you to see the difference between that, because this is a important way in which you can get things wrong when it comes to interpreting peptide sequences. So a tripeptide, A is the N-terminal amino acid, so we start with the N-terminus, which is an ammonium group, and then the alanine side chain is here. Amide bond to glutamate, so there's the nitrogen of glutamate, the carboxylate of glutamate. The glutamate side chain is alpha carbon, beta carbon, gamma carbon, and then carboxylate. Alpha, got to make sure I did that right. Alpha, beta, gamma. Oh, that's wrong. That's too long, isn't it? Okay, so this is A, this is E, and then the last amide bond takes us to our last amino acid residue at the C terminus. That's tyrosine, which should be a benzene ring and an OH group. Okay, that's AEY. Notice that the pink amino acid is on the N terminus is alanine. The orange amino acid in the middle is glutamate, and the blue amino acid at the C terminus is tyrosine. If you make the amino acid YEA, the Y is at the C terminus. The E is still in the middle. So that part at least is the same. And then the alanine, nope, got ahead of myself, there we go. And then the alanine is at the C terminus. Those are isomers of each other, but you can see they're definitely not the same thing. In the BA peptide, the Y is at the N terminus, and the A is at the C terminus, which is exactly the opposite from the AEY peptide. Yeah?
Can you explain again once again, or why the E is wedged instead of dashed?
When the nitrogen is on the left and the carboxylate's on the right and the alpha carbon is at a valley, it'll be a dash. Here it's at a peak, meaning that that amino acid got flipped over. So to keep the right stereochemistry, that's got to be a wedge rather than a dash. Yeah, so do you see what I'm trying to say with N to C? I mean, I feel like I'm disclosing my evil secrets, but I have definitely asked questions before. Which of the following is the peptide? Write a sequence. And guess what? One of the options is going to be for it to have the right letters but backwards in the structure. So you got to pay attention to where the N terminus is and where the C terminus is. What I'm saying is I could easily say which of these peptides is AEY, and I give you both of these options. So knowing the amino acids is not enough. You have to also know this rule that we always write out the sequences N to C. Okay, questions about that? So peptides can be hundreds of amino acids long. Yeah, go ahead.
Did you just choose those ones at random?
I did. Yep. I just chose them at random and then it happened to spell something.
But any could be used and the same will apply.
Any could be used and the same rule would apply. So on your problem set, for example, one of the questions this week is draw the peptide I like chem as it would appear in your blood. And please be aware of subliminal advertising. And if you want to spell out, I mean, if you want to practice your amino acids, try drawing peptides and spell out some interesting things from wherever. For example, Padme is dead might be one, or you could probably spell Anakin. If I told you about the amino acid ornithine, you could spell Kylo Ren or Yoda. Anyway, but yeah, one fun way to practice the amino acids, practice the structures, is to write a sentence. You have to use only the 20 letters that we have and see if you can spell some interesting things. Okay. So now I want to talk about proteolysis, which is what happens, and this proteolysis happens everywhere, but in particular in your stomach. And the enzyme that we'll talk about is called trypsin. And trypsin is an example of an enzyme that's called a serine protease. A protease is an enzyme that hydrolyzes amides in proteins to the corresponding carboxylate and ammonium group. So whereas we will later talk about how peptides and proteins are synthesized, now we're talking about degradation, which is going to be breaking the amide bonds in a peptide by adding water to liberate an ammonium group on one side and a carboxylate group on the other side. For example, if we start with our yay peptide, a protease would add water to break apart the amide bonds. So for example, suppose this protease broke apart the amide bond in the YEA peptide. What would that convert it to? Well, that would hydrolyze the bond between the alanine and the glutamate. You'd be left with a carboxylate on this side and an ammonium group on this side. If you compare the mass of what we started with the mass of what we ended up with, we added water to these molecules and they split apart. So that's a hydrolysis reaction. And then of course, if we did the same reaction again on the amide bond in the YE peptide using the protease enzyme and adding water to the molecule, that would hydrolyze the amide bond between Y and E. And on the E side, you would have just the ammonium group. And on the Y side, you would have the carboxylate. So proteolysis with these proteases hydrolyzes the amides into their respective amino acids. Questions about that? Now, how uphill or downhill do you expect that to be? We're breaking amide bonds, so let's focus in. What level of reactivity is an amide bond? It's 3. It's our one thing in the level 3 category. Good. So amide bonds are level 3. They get hydrolyzed to form carboxylates, which are level, and the new carboxylates, I guess the blue amino acid already had a carboxylate, so we're not going to count that. Let's count the new carboxylates, the yellow carboxylate and the pink carboxylate, which are all level 4. So amide hydrolysis is downhill in energy. Nevertheless, amides are stable enough that amide hydrolysis on its own is slow, which is why when you take a shower, your hair and skin cells don't break apart into the component amino acids and you melt, right? The amide bonds in your proteins are pretty stable. You need a catalyst to start chewing them apart. And in your stomach, you've got a catalyst, and that's trypsin and some other proteases. So what I want to do now is show you how trypsin proteases work. This is going to be a review because they work in exactly the same way that the lipase enzyme works. They're going to use the same catalytic triad. So catalytic triad in serine proteases like trypsin. So draw the amide bond between your, and this is one of the amide bonds in your molecule. So let's suppose this is the orange amino acid and this is the blue amino acid. And we're trying to hydrolyze this amide bond between the two of them. You're going to need a serine amino acid. And I don't know which amino acid it is in, I'll use the 1-letter code for serine. You're going to need a histidine amino acid, and I don't know which one it is in trypsin. There we go. And then, as we saw before, the histidine amino acid is going to be closely associated with a negatively charged aspartic aspartate residue. This is aspartate. You're looking at the beta carbon of histidine, the beta carbon of aspartate, and the beta carbon of serine. Not shown across the other side of the squiggly line is the alpha carbon and the backbone. So as you saw before with lipase, the hydrogen bonding of aspartate with histidine makes histidine more basic and more nucleophilic such that it can deprotonate serine, which then attacks the amide bond to give you a tetrahedral intermediate. As with the lipase, there's a oxyanion hole or some hydrogen bonding groups that stabilize the negative charge on that tetrahedral intermediate. So now we've got this tetrahedral intermediate. The histidine has an additional proton. It's now positively charged. but it's still hydrogen bonding with the negatively charged aspartate. Questions so far? Yeah. I showed you numbers in the problem set, or when we did lipase. No, you're not crazy at all. I'm trying to remember what day that was. I can't even remember the day. Not that day. That was citric acid cycle. Well, maybe that day, the 29th? Nope. The 30th? Yes. So this is quiz 23 from last Friday, or lecture 23. I showed numbers for the lipase. That's the number in the sequence of that protein. Is that what you're asking about?
Yes.
Okay. I happen to know them for lipase. I don't know them for trypsin. The number there refers to, if we were to write out the sequence of the lipase enzyme, it would be more than 263 amino acids long. And the number refers to the position of that particular histidine or serine in that long sequence. Yep. I just don't know what the numbers are for trypsin. Okay, so now we're going to kick off the amide, the nitrogen. That's the blue amino acid. The negatively charged oxygen is going to kick down and kick off the blue amino acid. And as the blue amino acid is leaving, it will pick up a proton from histidine, which will go back to its neutral state. That will lead to what we saw with the lipase enzyme. We're going to get an acyl enzyme intermediate. The blue amino acid, which I'm just going to represent by drawing the amine portion, is now free. It's been hydrolyzed off. The orange is still present in the active site, and now it's linked to that serine side chain. Which is still interacting with the aspartate. So, we're not done yet because we have to release the yellow amino acid and whatever it's linked to from the enzyme. And we're going to do this with water, using histidine as a base to deprotonate water. So, talk me through what's going to happen next. What do you think is the first step here? So, I'm hearing deprotonation. Where's my arrow going to start? From the histidine nitrogen lone pair, and then to what? One of the water hydrogens, then it's going to do what? Attack the carbonyl carbon, get a tetrahedral intermediate. Good. So we'll draw that tetrahedral intermediate. We'll also draw the histidine, which now is positively charged because it stole a proton from water. And it's still hydrogen bound to the nearby aspartic acid. That's a negative charge in the circle there. Sorry, it's a little bit crazy. Now what? I should have electrons on that oxygen kick down, and then who's going to leave? Serine from the enzyme. As it's leaving, it should pick up a proton from histidine. That gets rid of the orange amino acid as the carboxylate, or the carboxylic acid, and it can lose its proton due to the pH in a little bit. We won't worry about that. And your enzyme is restored to its starting state, where the serine is now liberated, it's neutral, it's ready to go again. The histidine is also neutral, ready to go again, and the aspartate is just in its same spot, hydrogen bonding with the histidine. We're now ready to go again with another amide bond. So if you're a peptide that's hundreds of residues long, this enzyme has to bind here and clip this amide bond, and then it's going to bind here and clip this amide bond, and here and clip this amide bond. You've got a lot of amide bonds to hydrolyze. Some enzymes like trypsin prefer specific amide bonds. I need to look this up, but if we looked it up, trypsin would prefer to cleave amide bonds between a certain amino acid on the left and maybe a variation of amino acids on the right. So your body makes tons of proteases that can clip all the amide bonds. In your stomach and intestines, this process happens. You first make shorter peptides, and then those get cleaved to smaller peptides and smaller amino acids. Yes?
Could you give us a 30-second summary of the most important stuff you want us to maintain from today?
30-second summary, the most important stuff. Wow, yes, we should just ask ChatGPT, but instead I will say 20 amino acids, their names, 3-letter codes, 1-letter codes, structures, and have a feel for what category they belong in and what their major properties are. And then from this last part about peptides, N to C is how the sequence is written. And then for this part about amide bond hydrolysis, how to apply what you learned about the catalytic triad to draw a mechanism for hydrolyzing amides. Okay, that's it for today. Thank you. Don't forget to pick up the problem set, and I will see you next time.